Today we're going to be talking about the fascinating process of protein synthesis. Now, you may be wondering, what is protein synthesis? Protein synthesis is the process of taking a chunk of DNA and turning it into a chain of proteins through a whole bunch of different steps. Now, these steps can be categorized into two major parts called transcription and translation. Part 1. Transcription. The first step in protein synthesis is to have our strand of DNA we would like to copy be unzipped by an enzyme called helicase. Without helicase, DNA would keep its double helix or wrapped structure, which would make it impossible for our next enzyme, RNA polymerase, to craft a complementary strand of mRNA by pairing up the matching nucleotides and then separating from the DNA, making a whole new strand to send out of the nucleus of the cell and into the cytoplasm. It's important to note that on our strand of mRNA, there are these things called codons. Codons are just series of 3 nucleotides within the strand of mRNA. These codons can be translated into different amino acids, which will be what makes up our final chain of proteins. Now that mRNA is outside of the nucleus, it gets surrounded by organelles called ribosomes. This is where our first part ends and our second part begins. Part 2. Translation. So our good pal mRNA is now surrounded by ribosomes. These ribosomes bind to the nucleotides of our strand of mRNA, beginning at the start codon, which then initiates protein synthesis. We should note here that there is something called tRNA, which is a molecule linked to an amino acid. The codons on the strand of mRNA let this tRNA know which amino acid it needs to carry over in order to bind with the mRNA. Each molecule of tRNA has something called an anticodon attached to it. This is just the matching set, if you will, that will pair with each codon on the strand of mRNA. These must bond because it ensures that the correct amino acid gets where it needs to go. These amino acids are important because peptide bonds form between them, which makes a polypeptide chain, which is a fancy term for a long chain of proteins. This will go on for the entire length of the strand of mRNA until it hits the stop codon, in which the process of protein synthesis is complete. But why is this important? The creation of proteins is necessary to any living organism's survival because proteins are responsible for all cell structure and function, so protein synthesis is a crucial part of what goes into the making of our cells.